It's my pleasure to introduce Dr. Kelley Kidwell, a professor of biostatistics from University of Michigan School of Public Health. And her specific methodological focus is on sequential multiple assignment or randomized design or SMART designs. And so thank you for being here, and thank you for the presentation.
Yeah, thank you so much for having me, and thanks to everyone who is taking their Friday afternoon to listen to all of us today. So I'm going to talk about SMART design, or sequential multiple assignment randomized trials. But before I do so, I want to motivate this type of clinical trial by talking about what we call dynamic treatment regimens. So for most illnesses, conditions, diseases, whether they're chronic or acute, often treatment is not just a one-time thing, right? You don't usually just present yourself, get one treatment, and you're good to go. That would be amazing if all conditions were like that. But often what happens is that you have some condition, you go to a clinician, they give you some treatment, and then and they're deciding which treatment to give you based upon some information they've gathered about you and about your condition. And then they provide that treatment and they check up with you. So this is an example of treating depression in adults where something like that is happening, where the clinician is thinking about what's your history, what is the level of depression, severity of depression, and what's happened in the past, and based on that, deciding which type of treatment to give, and then checking up and deciding whether that needs to be revised, and what needs to happen as you move along in the treatment. So we can term these sort of treatment strategies dynamic treatment regimens. That's the statistical terminology that we use, but you might have heard them called adaptive interventions or stepped care or treatment policies. They're all the same thing. And they're just this sequence of individually tailored decision rules that specify whether, how, and or when to alter the intensity, type, dose, or delivery of the intervention at these critical decision points over the course of care. We can also use these dynamic treatment regimens thinking about education interventions or implementation science. How do we get evidence-based practices into clinics or different places? But today I'm going to focus mostly on sort of clinical care. So really, this fancy terminology of dynamic treatment regimen, or I'm going to keep referring to it as DTR, is just this evidence-based guideline or formula for the intervention that we want to give the clinician at the beginning of care. And so that they have this strategy in mind when treating a patient. What do we do first and what do we do next, depending upon how they did on what happened first. So we're really just trying to operationalize this decision-making that occurs in practice. We really want to get evidence behind the dynamic treatment regimen instead of just being based on sort of clinical experience, which can be great, but can sort of be a black box. So we want to unpack that black box and figure out what is it that are actually the active parts of the interventions. And so we'll see how to do that via SMART design soon. But let me give you an example of a dynamic treatment regimen. So this is one dynamic treatment regimen for treating pain for women with breast cancer. So for individuals with stage 1 through 3C breast cancer experiencing pain, give 5 one-hour-long sessions of pain coping skills therapy. At the end of these sessions, reassess pain. If the pain score has decreased by at least 30%, then there's no further treatment. But if the pain has not decreased by that threshold, then we'll provide 2 additional pain coping skills therapy sessions. So this is one dynamic treatment regimen that describes all the scenarios for all the patients given these different responses to the treatment, right? So one person isn't going to follow this whole protocol necessarily, but they're going to follow pieces of it. What do they get first? And then depending upon how they respond, what do they get subsequently? But since we want to know this regimen up front, we have to know what to do if they do respond and what to do if they don't respond. And as you can see and imagine, since we're all patients at some point, right, these dynamic treatment regimens can be incredibly useful when diseases or disorders or conditions wax and wane. So we might have to be more aggressive up front and then we can back off, or we could be maybe sort of less aggressive and then intensify later. Similarly, when there's no widely effective intervention, right? What works on average may not work for everyone. And so we might have to modify or adapt that treatment based upon how individuals are doing. Similarly, as I just mentioned, you might want to start with something less burdensome or less costly and only ramp up if necessary. Or perhaps you really have to, go aggressive and then can ramp down. And if there are adherence problems such that you try something and individuals aren't adhering, you want to be able to know how to modify based on that adherence. And of course, what works for me today may not work for me tomorrow, or what works for me may not work for you. And so we want to have that ability to know what to do next. And these dynamic treatment regimens are going to give evidence-based treatment strategies for that course of care. However, right, there's many pieces in this dynamic treatment regimen, right? And there's lots of questions then that go along with it. How do we get the evidence for what's best to start with? How do we understand when and how we measure that level of response to that initial treatment? And then what do we do for people who seem to be doing well? And what do we do for people who seem to not be doing well, right? We want to unpack that black box and figure out, well, how do we put together optimal components to come up with this really robust dynamic treatment regimen? And so one way to do that is using a clinical trial, which is called a sequential multiple assignment randomized trial, or a SMART. So a SMART is just a multi-stage clinical trial where each stage of that SMART design is going to correspond to a critical decision point. Like, what do we do first? Okay, and then at the second stage, well, what do we do subsequently for responders or non-responders? And a randomization is going to take place at each of these critical decision points so that we can answer these questions without issues of observational data. So without the potential confounders or selection bias, et cetera. What we want is for every individual in the trial, they're going to move throughout all of the stages of the SMART design. Our goal, or the motivation behind running a SMART design, is really to inform the construction of, or to estimate the effects of, or to compare these embedded dynamic treatment regimens. So here's an example of a SMART design. This is one potential option where individuals are randomized between two initial treatments, A and B. They're followed for some amount of time, and then their response to that initial treatment is assessed. And in this design, the responders to treatment only have one option. So they're assigned the next treatment. That might be to continue or perhaps discontinue that treatment. And it can differ depending upon your initial treatment. In this SMART design, non-responders to initial treatment are re-randomized between different treatment options. These are usually things like augment the treatment or switch treatment or intensify treatment. And again, these could either be the same options in the second stage for each of the initial treatments, or they could be different. And so you can tell this is a SMART design because there's two randomizations in sequence. Everyone in the trial doesn't have to be randomized twice, but some subgroup of participants needs to be randomized in sequence for this to be a SMART design. We can also see that it's a SMART design because that subsequent randomization is restricted based upon response to the initial treatment. And so it's not just a crossover, for example, right? Everyone's not getting something else. We're deciding what to do based on the response to initial treatment. That randomization within the SMART is acting the same way in a SMART as it would in any clinical trial, right? This randomization is going to allow for valid causal inferences about the effectiveness of the treatments involved. I'm going to say effectiveness because usually these SMARTs consider all active treatments. We don't usually have placebo in SMART designs. We're not trying to find the efficacy of treatments. Rather, we're trying to find the effectiveness of these different sequences and dynamic sequences of treatments. They're also going to provide the basis, this randomization provides the basis for identifying important individualization or further tailoring variables at each of these critical decision points. So this is akin to, like, in standard one-stage trials, what subgroups of individuals does each treatment work best for, right? These tailoring variables we can find at the first stage and the second stage to figure out which subgroups of individuals are these sequences best for. And one note, right, the clinical trial is the SMART and includes randomization. But the result of the trial, what we want are these estimates of the dynamic treatment regimens. Those are the treatment guidelines. Those don't include randomization, right? Those are the formulas or the guidelines of treatment. First start with this, and if you respond, get that. And if you don't, get this other thing. So whenever we talk about dynamic treatment regimens, those aren't including randomization. We're just using randomization as a tool in the trial to get those unbiased estimates. Here's another example of a SMART design. So this differs a bit from that first example that I showed you because here the responders are re-randomized as well as the non-responders. So again, there are 2 treatments initially, and then responders are randomized between 2 treatments. It might be continue or discontinue. And non-responders are re-randomized, for example, augment or switch. I've listed C, D, E, and F in the second stage, the same for those who got treatment A and who got treatment B, but they could be different, right? The way that you figure out which treatments to include are what's actually being used in clinical care, right? The design is highly motivated by what's being done in clinical care and trying to say, this is how people are practicing. How do we actually find out if those are the most effective dynamic treatment regimens? So within this SMART design, we can talk about how the dynamic treatment regimens are embedded within here. Now we'll see that, if we follow these arrows, one patient, right, one participant will follow one of these pathways, right? And there are 8 pathways. But there are also a number of DTRs, right? And remember, the DTRs include 2 pathways. So it's always going to include a pathway for responders, an initial treatment, a response, and their second stage treatment. And the DTR will include that initial treatment with the non-response and the subsequent treatment. So for example, one of the embedded dynamic treatment regimens in this SMART design, we can say from this treatment triplet, first receive A, if you respond, receive C, and if you don't respond, receive E. That's one dynamic treatment regimen. Another dynamic treatment regimen is now highlighted in yellow, where first receive A, if you respond, receive C, and if you don't respond, receive treatment F. So there are 8 pathways, but we can see that every DTR includes 2 pathways. In this case, there are 8 dynamic treatment regimens. But again, don't be confused by the number of pathways. It's really the combination of pathways that leads us to these DTRs. So there are 4 DTRs that start with treatment A and have a pathway for response and non-response. And there are 4 DTRs that start with B and have a pathway both for response and non-response. So I've listed those treatment triplets, each one of these being a DTR embedded in this SMART design. If I go back to the first SMART example where the responders all were assigned the same treatment, they weren't re-randomized, we can see here's an example where the number of pathways does not match the number of DTRs, right? Each participant would follow one of these 6 pathways. However, what we're really interested in are these 4 embedded DTRs. Is it better to start with A, and if you respond, continue with C, or if you don't respond, get E? Or is it best to start with B, and if you respond, get C, and if you don't, get F, right? Those are 2 of the embedded DTRs that might be motivating this SMART design. I want to show you one more SMART design. I could take up the whole time. You could be here till 6 p.m. or after with all the different types and variations of SMART design if they allowed me, but don't worry, I'm not doing that. SMART designs, right, can have any number of stages. They can have any number of treatments at the first and second stage. Usually this response is some binary variable, but it doesn't have to be. So as you can imagine, there are lots of different varieties of different SMART designs. What I'm showing you are the 3 most commonly used SMART designs that are 2 stages. And this one differs a bit because whatever we did up here for treatment A, we didn't have to do the same thing for treatment B. So this design has been used in particular when B is like a more intense or resource intensive or burdensome treatment. And so there aren't as many options to follow up with. So in this case, right, people who first get B, they're not re-randomized again. But this is still SMART because there is a subset for which there is sequential randomization. So in this SMART design, There are 5 treatment pathways, so any individual would follow one of these treatment pathways in the design. But what we're really interested in is figuring out the effects, estimating the effects of these 3 dynamic treatment regimens. 2 that start with A and include, both of these include that same subgroup of first receive A, and if you respond, receive C, but they differ by what to do for the non-responders. And then there's 1 dynamic treatment regimen that starts with B, and it's right, we have no randomization in here. This is just the one DTR option for those who start with B. So you might be thinking, oh, I know, like this design looks really familiar to me. It might be familiar looking because smarts have existed over the last few decades. A lot of the literature came out in the early 2000s and has been really booming since. And if you're not familiar with smarts, you might be thinking, oh, this really looks similar to a factorial design, right? A factorial design is where you could have, if you're interested in 2 treatments and they each have different levels. So like you have treatment A and you have maybe an intense and a less intense version of it, and you have treatment B and you have an intense and a less intensive version of it. You can do that 2x2 table where you have all the cells of like both the intense versions of A and B, one intense A, low intense B, low intense A, or low intensive of both of those, right? That would be a factorial design where you're looking at those combinations. A SMART can be very similar to a factorial design, but it's in sequence. And so you're not going to necessarily, right, get those treatments at the same time, but you're going to see what happens if you sequence them. Often in factorial designs, while we might be interested in the treatment interactions, we often assume that they aren't interactions so that we can get this gain in power from using the design. But really in smart designs, we're highly motivated to explore those treatment interactions or potential interactions between the initial treatment and the second stage treatment. So it differs a bit there. I've also noticed that a lot of people want to put smarts into the box of adaptive design because we talk about adaptive interventions or adapting the treatment for individuals based on their response. But I think it's really important to note here that usually when we use the word adaptive design, we're referring to between patient adaptations such that we're using accumulated data in the trial to make decisions to change certain operating characteristics of the trial for future patients. That's using, some patient data to affect what the trial looks like for future patients. Whereas in the SMART, we're usually not doing that, right? There are adaptive SMART designs, but generally the standard SMART is not adaptive, that the within patient, their treatment can change based upon their response or non-response. But all of this has fixed operating characteristics within the trial and is not affected by another patient's data. Finally, this may look similar to a crossover design, but smarts differ from crossovers for a couple of reasons, one of which is we don't want washout periods in a smart design. Again, we're interested in those treatment interactions. And another thing is that in a crossover design, everyone must cross over to the subsequent treatment. Whereas in a SMART, we have restricted crossover usually, where responders, for example, may not cross over. They may continue, or they might discontinue, and non-responders would continue. And this could probably help with retention in the trial, both retention and adherence in the trial. So why use a smart design? Well, you would use a smart design because you're highly interested in these sequences of treatments. So you're in any clinical trial design, you're motivated by the science and the design matches the science. You don't first get motivated by the design and then try to match it to the science. And so you're trying to think about, how do I understand what are these best dynamic treatment regimens? And smarts are incredibly powerful because they can answer many questions regarding these dynamic treatment regimens. But it's important to note that one SMART can't answer all these questions. But for example, a SMART can think about, well, what do you start with? Should you start with A or should you start with B? What do you have subsequently, and do those things interact? Is there something where the first treatment really sets you up for a better response on the second treatment, or the second treatment really has some positive synergy with that first treatment? Also, do you learn information from the first treatment that can better set you up for selecting the second treatment? Additionally, you might have questions about, I know that we need to adapt treatment, but I'm not sure when, or I'm not sure how best to decide, what to measure to adapt that treatment. So you can have these questions about what we call that intermediate tailoring variable that I've been referring to as response. Well, when do you measure response, or how do you measure response? Smarts can be done to look into those questions as well. And then subsequently, you can also be thinking about, well, what are these specific subgroups for which these sequences of treatments work best for? So what are these additional tailoring variables at baseline and along the way to second stage randomization that I may figure out these specific subgroups of individuals, this dynamic treatment regimen is going to work best for them. So smarts can be incredibly powerful clinical trial tools to address these questions about dynamic treatment regimens. Since you can't answer all those questions at once, right, we really need to make some critical decisions about, well, which one do we address? And again, this is like any other clinical trial where you want to think about your primary aim in terms of, well, what decisions are most controversial or, most need investigation? Well, what decisions are likely to have the biggest impact on clinical care or on the outcome? And so those would be, really how you dive into your primary aim, and then how you think about the sample size and the primary analysis of your SMART design. There have been a number of SMARTs in the field. They sort of started off in oncology, given the natural sequencing of treatment that occurs in treating cancer. But there's been an explosion of SMART designs over the last 2 decades in many different areas, particularly in those using both pharmacologic and non-pharmacologic treatments. So instead of usually these trials are looking at, maybe one drug, but multiple behavioral or non-pharmacological interventions and how they best go together. So you can see lots of mental health areas, but they've also been used in education strategies, and some prevention in terms of like smoking cessation or cancer prevention, suicide prevention. And then also they've been used more recently in sort of a cluster way in implementation science of how to embed evidence-based practices in practice. There's a really great website that's from Michigan and the D3 lab led by Danny Almiral and Imbal Nahum Shani, that if you click on there, you can see a really awesome list of SMARTs that have been in the field. I should also mention that these SMARTs have been funded by NIH, PCORI, and also DOD. So there's lots of interest in these types of designs. But again, the design should be motivated by the questions at hand. So the best way to learn about a clinical trial design, I think, is to really see an example. So this is a recent SMART that ran for the behavioral cancer pain intervention dosing, run from Tamara Summers at Duke, and also included Eric Laber on this group, who's been amazing in the SMART space in terms of methods. I have both the clinical trials information here, but also what's really awesome about the SMART is they have both a protocol paper and a primary results paper. So you can really see how you could think about the protocol for a SMART, and then how you would think about presenting the results. So this is to look at different doses of this behavioral intervention called pain coping skills training, and then what to do best after an initial dose level of this training. And so you'll see that the initial interventions are either a full version, which are 5 sessions, or a brief intervention, which is 1 session. So I took this from the protocol paper. I apologize, it's a little blurry, but cancer patients with higher pain scores are randomized between the full session or a brief session. And then after I think it's between weeks 5 and 8, I'll have it on the next slide. After these sessions could occur, they look to see if they have a decrease in pain. And if they don't, regardless of the first stage treatment, they're randomized between essentially like, an intensification of that initial treatment, or a maintenance version of that initial treatment. So it differs a little depending upon what you got first, but it's the same idea of an intensification or a maintenance. And if they did respond, then they're randomized between a maintenance version of that initial treatment versus no further intervention. And then subsequently, they had a few follow-up times to which their pain was assessed after the interventions. In any smart design, you got to show the design, and then you have to show this table, or you should show this table of the embedded treatment sequences or dynamic treatment regimens. So they call them sequences instead of getting bogged down in the DTR vocabulary. And I love what they did here, and I try to tell anyone interested in a smart to do something similar. Well, you'll call out what that sequence is, or that DTR, right? This was like our second example, so it has 8 embedded dynamic treatment regimens. You list the first stage treatment, and then what happens if they respond and what happens if they don't respond. Now, part of the reason why they use this sequence terminology, one, because maybe DTRs doesn't really resonate with clinicians or people who would read this, but two, because you'll notice that 2 of these strategies aren't dynamic. So these 2 strategies, S3 and S6 or S7, say, you know, everybody start with this full version of the pain coping skills training, and you know, after that, everybody gets the full maintenance. So it doesn't matter whether you responded or not, right? It's a static regimen. It's not dynamic. And similarly down here, everybody get the brief version, and then subsequently, everybody get the brief maintenance. All of the 6 other strategies are dynamic, meaning that the second stage treatment depends on response. But for these 2, they're static regimens or sequences. So something else that immediately people, when I show the smart design, people are like, oh my gosh, you're going to need thousands of patients to run one of these. But fear not, you do not. As in any trial, the sample size is dependent upon the primary aim, which can differ vastly in a smart design. So in this trial, it was based on a total sample size of 327 patients, and I'll show you how they got to that. Again, I've just listed the stage 1 treatments and the stage 2 treatments. Their tailoring variable, or that measure of response, was measured between weeks 5 to 8 and was a pain assessment based on their brief pain inventory pain intensity score. And it was a change in the score from baseline to that week 5 to 8 time point. Their outcome was similarly this % change from baseline in the brief pain inventory. We can have discussions on, you know, % changes as outcomes or whatever subsequently, but we'll just, you know, this is what they used. So here's how they wrote their hypotheses, and these are very common hypotheses for smart design. So again, this is a reason why I chose this as an example. I'm not the statistician on this trial, but it's a phenomenal example because of the literature that exists and because it really exemplifies what often people do with smart designs. So the primary hypothesis was really a 2-arm trial hypothesis. It was as if you just did the first stage. And so the first primary hypothesis said, you know, is getting this full version of the intervention better than getting a brief version in terms of the change in pain from baseline to that week 5 to 8? So they looked at the end of the first stage, which one was better. Now, you might think, why are they doing a smart when this is the primary reason? They could have just done that, right? Well, a few reasons. One, clearly this question hadn't been answered yet. And so great, we get to answer it in this smart. 2, not only are we going to answer this question, right, we're going to get to also explore all of these other different questions regarding the dynamic treatment regimen. So we get so much more from this one trial than had we just stopped there. They also, as a secondary question, asked, well, at the end of the trial, right, is it best to start with the full sessions or start with the brief sessions? And that's going to average over everything that happens subsequently. Now, this sometimes is a primary aim in a smart. And one, it's nice because it boils down to a sort of standard 2-sample question. But 2, you might be thinking that's not really a relevant question because ultimately you're going to figure out what to do subsequently and not average over that. And so that's why I think they went with, right, just at the end of the first stage, let's compare it. This is a little bit less clean because hopefully we learn what's best to do and we implement that in practice. Okay, so sample size, you don't need special things here, right? You can just use a 2-sample t-test. In fact, that's what they did. Here are their parameters. They wanted to find a 10% difference between the two interventions in terms of the percent change in pain, and they accounted for 15% attrition. Now, if, however, you happen to have other types of questions, particularly regarding the dynamic treatment regimens, and wanted to size that trial, we have a really beautiful R-Shiny sample size calculator for the 3 most commonly used SMART designs. So I'm happy to share these slides. Also, the link is actually written out on the next slide. You can click on this, and then you'll click on the design that you're interested in, and then you'll see that whether your outcome is binary or continuous, or if you have a primary aim regarding the first stage treatments or the second stage treatments, or the dynamic treatment regimens, this sample size calculator can help calculate the sample size required. And so it's this nice interactive R-Shiny applet that will highlight what it thinks you're telling it to power on to ensure that's the case. And it will give you results below, which you can inflate based on missingness or potential loss to follow-up. Okay, so what happened in this trial, right? We actually have the results. And so we can see their primary aim. What they found was that there was a difference between the 2 initial treatments in the change in pain of nearly 14%. And that was a statistically significant difference at the 5% significance level, such that it really did seem like the pain coping skills training full 5 sessions was more effective in reducing pain than that brief only receiving 1 session. So what was their second aim, or what could be a second aim in a SMART design, right? Well, we had a SMART, we had all this re-randomization. We really want to attack that, we want to be able to both estimate and possibly compare those embedded dynamic treatment regimens. And so when you do stage-specific analyses, right, that's going to look really standard. You can use standard tests, regression, whatever you want, right, for the power and for the analysis. Once you get to thinking about the dynamic treatment regimens, you usually need SMART specific analyses or power calculations. And so what they plan to use, which is probably the most commonly used analytic method for SMART designs of a either a longitudinal or a cross-sectional analysis, is what we call weighted and replicated regression. And this is just going to use generalized estimating equations to analyze the effects of the, in our model, we're going to put the first stage treatment, the second stage treatment, and their interaction. I'll show you that model in a second. They were specifically interested in looking at the comparison of the most intensive versus the least intensive approaches. So those S8 and S1, that refers back to that initial table I showed you of their strategies. So very briefly, because I'm already going to talk longer than I should, if you want to learn more, I'm happy to point you to the references. But weighted and replicated regression is where we're going to replicate the data to deal with the fact that some people are part of really multiple DTRs. Like, so for example, right, if first receive A, and if you respond, receive C, and if you don't respond, receive E, right? The people who respond and get C, those individuals who follow that pathway are consistent with that DTR that I just noted, and also one where if you first get A and then get C, if you respond, or get treatment F if you don't respond, right? They're used in both of those DTRs. And so if we want to simultaneously estimate the effects of both of those DTRs, we need to use their data twice. And so we'll replicate them as if they're in that data twice, but then we have to say, we're basically just trying to trick the software to use their data twice, but we need to account for the fact that they're, the same person, right? They're not more data. And so we'll do that. And then the SMART design, sometimes we only re-randomize specific groups of participants. And so in order to account for any imbalance by design, we'll need to weight the data by their inverse probability of receiving that treatment or being in that treatment pathway. In our SMART design, or in this example here, right, everyone's re-randomized. So if we use the known probability of randomization, those weights would all be the same. But in other smart designs where it's only the non-responders are re-randomized, the weights for responders and non-responders would differ. So we'll replicate the data to trick the software to use their data multiple times. We'll have to weight the data if we have any imbalance by design. We could also weight the data by the empirical proportions for a bit more efficiency, and to account for any sample-to-sample difference in weights and the fact that these people aren't extra people, we'll use the robust or sandwich errors on the generalized estimating equation for the correct standard errors on our effects. But this can all be done using standard functions in R. So for example, this is a saturated model where O could be any baseline covariates, for example, like we just learned are so important, our stratification variables, and our first stage treatment, our second stage treatment for responders, our second stage treatment for non-responders, and their interactions. Now, this model depends on our smart design, so it will differ if our smart design looks different. And here's what they found in that example SMART for pain coping skills in the intervention. So again, we can see here, these were the change in pain scores from baseline to the end of the first stage. So they're just listed, there's just two of them because there are two first stage treatments. Then at the end of the second stage, so this is immediately post the second stage intervention, we have the estimates of the pain coping scores for each dynamic treatment regimen or each treatment sequence. And then this A4 is a subsequent 6 months later, so a longer term post-intervention follow-up. And what they found was that at the end, the initial, just at the end of stage 2, there was no significant difference between all of these dynamic treatment regimens. But at the end, 6 months later, they did find a significant difference between these dynamic treatment regimens. And what you can see is for those that got that 5 full sessions initially, they tended to have more reduced pain longer term than those who only got the brief session initially. They did compare the most intensive versus the least intensive versus some walled tests, and they found that there are no significant differences between these dynamic treatment regimens. Both, when I say significant, I mean clinically significant and also statistically significant. Then after you've had your primary aim that might deal with the first or second stage comparison, secondary aim, which deals with your DTRs, often an exploratory aim has to do with finding those subgroups for which the DTRs are most effective, or what we call like further tailoring of the DTRs or further personalization of these dynamic treatment regimens. And so those are really looking at different moderators of the treatment, and they explained that they wanted to evaluate the potential improvement in pain reduction that could have been realized for each participant if she had received the initial and secondary interventions via the optimal treatment strategy. So what we're really looking for in these analyses are baseline demographic and disease characteristic interactions with the initial treatment, and also baseline and any characteristic up to the time of second stage treatment assignment interactions with that second stage treatment assignment. And for these types of analyses, usually what's used is what we call Q-learning, although there are a host of different options here. And this is essentially just a form of backward regression to account for the fact that we have sequential treatment. And it will, I won't go into the details, but I will say that an R package exists to do Q-learning. It's called DIDACT Regime. And there's also a really awesome resource that is a website from Eric Labor's lab that goes along with his book with Marie Davidian and Butch Ciatis about smart and dynamic treatment regimens. And it goes through how to, use Q-learning in this type of setting. And so I have an example here of what we'd be looking for, like what variables were associated with a DTR that looked optimal for individuals. Okay, so in summary, I know that was a flyby, very quick introduction to smart design. I'm happy to take any questions or follow up via email, but essentially, SMART design is a clinical trial that's a multi-stage clinical trial where some set of individuals are randomized at least twice in sequence. And these different stages correspond to critical decision points over the course of care. And often subsequent treatment assignment is based upon some tailoring variable like response to the previous treatment. SMARTs are highly motivated by providing rigorous evidence for dynamic treatment regimens and can capture these treatment synergies or antagonisms or delayed effects between the first and second stage or further stage treatments. If you're doing stage-specific analyses, power analyses, or analysis of the data, you can use standard methods. If you're doing sort of something having to do with the dynamic treatment regimens, usually you need these SMART specific methods that will consider the multi-stage part of the trial and also the fact that some people are consistent with more than one DTR. But there are multiple R packages that can help with both power and analysis, many of which can be found on that D3 website. And there's also a method for how to deal with missing data in a SMART design. So I list some resources here, the D3 website being a great resource for all things SMART. A few intro articles that I like to pass along to collaborators or clinical investigators who are really interested in SMART design. These are really accessible. They're not super mathematical and show examples. And so I think they're the best, sort of 3 intro articles that exist. And then there are several textbooks. I think this one, the Dynamic Treatment Regimes, is a really great one that has that corresponding lab website for help on it with our code as well. But these are both more statistical heavy, so for the statisticians, not for the clinical investigators. With that, I'm happy to take questions and also any follow-up at kidwell@umich.edu. And if you're interested in really taking a deep dive into SMART designs, I'm part of a group that does SMART-workshops. It doesn't stand for Sequential Multiple Assignment Randomized Trials. It stands for Statistics, Methods, and Research Training. And so you can check out that website for a lot more recorded and also eventually live workshops of interest. Thank you so much.
Thank you so much, Kelley. This was very informative, very, very interesting. And so there is one question in the chat, but what maybe first, if I can ask you a question first.
Yeah.
What do you see? I know you said there is an explosion of application in different areas, and it sounds very applied and very care related or very practical, depending on where the application is. Where do you see a barrier that it's maybe you would think it's so practical that would maybe even more common, but do you see any barriers to implementing this type of designs?
Absolutely. And I think that the largest barrier is in trying to sequence multiple drugs. And so even though this design was sort of originally used in oncology, it ended up answering stage-specific questions, not looking at the full regimens, which were really of interest. And I think it's one, if you have multiple drugs, how do you get multiple drug companies to be a part of it? But 2, the bigger barrier is FDA doesn't know how to register a regimen. And so there's lots of questions at the FDA level of like, well, what do we do with this information? Now they're getting more and more interested. There's been several workshops lately that I was a part of, particularly in oncology, of thinking about neoadjuvant and adjuvant, the neoadjuvant and adjuvant setting and how to think about that full regimen. So they're open, but they haven't yet had an example. And so someone needs to come through with that and be successful in order for others to use it.
Absolutely. Yeah, that's very interesting. So if we go back to the questions, so there is a question about how would this be different from phase 2 to 3 study? Or the question really is it done generally as a phase 2 to 3 study?
Yeah, this is a great question. So one, it's not often used in investigational new drug proposals yet, again, because the FDA doesn't quite know what to do with the regimen. But it could could potentially be on a stage specific basis, or, you know, to break that barrier. Often it's used in sort of, I guess it's more in this like phase 4 space where you have active treatments and you're trying to combine them and see, you know, the combination or sequence of treatments. And so, you know, we think about this of, we have these treatments that we know are efficacious. How do we put them together or compare them to other ones to see the effectiveness of it? And so they're used more with that. They're also used within smart designs, can be thought of as confirmatory or as more exploratory. If anyone's familiar with the multi-optimization strategy framework or the MOST framework that came out from Linda Collins and Susan Murphy a while ago. Actually, I have a slide on this, so I can maybe share it, which would be helpful. Let me go back to sharing this one. This is the MOST framework, and it's really about developing and or evaluating the components of any multi-component intervention. And so there's the MOST framework is, there's some preparation phase, and then there's some optimization phase, and that's where the SMART can come into play, but there are also other types of trials that could be used in this setting. That's trying to figure out what are the optimal components of the multi-component intervention. And then you might iterate on that for a while. And then you can go into this confirmatory or evaluation phase, which, for example, if you used a SMART, that might have been powered on the first stage treatments, but then you take sort of what looked like the most promising dynamic treatment regimen, and you put it in this confirmatory trial of a more standard 2-arm trial of the best, you know, dynamic treatment regimen versus standard of care, and you could evaluate it that way.
Right. Yeah, that's very helpful too, in terms of thinking where the sequence of testing certain regimes, right?
Yeah, so you can think, and you can run a pilot SMART up to, you can run a very large SMART, right? That's much more confirmatory on the DTRs. So lots of options.
We have a hand raised question
That's
Oh
right.
yeah.
for
Yes,
Kelley.
let me unmute.
NASANet, we're unmuting you. We hope. There we go.
Able to unmute now. Looks like you're unmuted, but we're not hearing you.
Oh, accidental raising of the hand. No problem. There's another question. Can it be done for the same IP where the second randomization is for repeated treatment or not? Yeah, so I think you're saying, right, like so you could have some initial treatment and then re-randomize perhaps responders or non-responders to receive that treatment again. I think if that's what would be done in practice, absolutely. But if that wouldn't be ethical or ever considered in practice because they've already shown to not respond or to respond to it, then you wouldn't want to include that in your trial design.
Okay, you got another question. No, I'm sorry. That's in
Oh,
the
I think, yeah.
chat.
I think Kelley, you showed one where there was part of, well, maybe without randomizing, continue the same strategy for the responders, but
Yeah.
for non-responders, maybe you can use the same, same treatment, but adjust the dose or adjust the method of administration.
Yeah, that's right. Yeah, that's true. Right. You could intensify the same treatment in the second stage.
Actually, we do. Frank Harrell has a question for you, Kelley.
Yeah, Frank, go ahead.
Hey, Kelley, in the old days, we had multi-period randomized crossover studies, and people tended to use those for acute diseases with short-acting treatments. Asthma would be a key example of an area where a lot of crossover studies were done. Are SMART trials really restricted to very short-acting treatments in more acute disease, or how far can you extend it?
I don't think so, but obviously you still need the SMART to be done within a funding / relevant amount of time. So in that sense, you usually, I would say you probably don't want each stage to be longer than a year. for all of that to be done. But you don't have to wait for a full effect to be seen on the first stage treatment to go into the second stage. So usually what you want to do is to be able to see some effect if it's there from the first stage, but not wait too long such that if it's not effective, you've completely lost the participant, right? So in that case, you might have this, it's really an intermediate outcome for which you're tailoring that second stage on, not a full-fledged outcome. So you could wait a little bit longer. It doesn't have to be super fast, but in the same realm, you need to have answers probably within a reasonable amount of time, within 5 years. If you're going to run hundreds of patients through it, probably can't be too long.
Thanks. And maybe I'll ask one more quick one. Have Bayesian methods been developed in this domain, and how do they compare, if so?
Yeah, the predominant literature in smart design is frequentist. However, there has been a increased interest in developing methods, Bayesian methods in this space as well. And actually, I guess I should say that a lot of these trial designs came from Peter Thal down at MD Anderson. And so many of his methods are Bayesian, and then sort of his progeny and others have developed some methods. A lot of the Bayesian methods, I would say, that exist thus far are really thinking about the further tailoring of the dynamic treatment regimens, as opposed to maybe the primary aims of the smart design. But I have just more recently been seeing some things about adaptive design in smarts that are using Bayesian methods. And then obviously they just, are different than the frequentist methods in terms of prior distributions and more computationally intensive, but can be quite interesting. I've also
Thanks.
myself have been using Bayesian methods with smart designs, but in small samples for a totally different perspective of using the second stage data to get more efficient first stage estimates. But in that case, it's kind of more like a crossover where you want a washout period. It's a very different setting. We call them small sample smarts, and I've now just confused everyone most likely, but it's quite interesting, and that's a great place for Bayesian methods.
Thanks.
Yeah.
Okay.
Yeah, there's just one more question that I'll address is insights if funding agencies like NIMH or NIH support smarts. They absolutely do. So I'm a co-investigator on over 7 smarts funded by NIH in particular. And I also know of several smarts that have been funded by PCORI. The smarts I'm a part of have been funded by NIMH and NIDA. But I think other agencies within NIH are also interested in smart designs.
Great. Thank you.
Great,
And
thank you.
I think that last question is actually very important for everybody.